Okay. Can you guys hear me?
Yes.
Awesome. All right, I just got to download chapter 13 real quick. Okay, while I'm doing this, anybody have any questions about what we covered so far? Okay, so this is chapter 13. And just to remind you guys, so the central dogma is a term that was coined by Watson and Crick when they discovered that DNA was a double helix. So just a reminder that the central dogma is that your cells contain DNA, and all living things, that, as long as, you know, from what we know so far, all living things have DNA. Viruses don't count because they're not considered living because they don't meet all those criteria that we defined in chapter 1. So DNA is converted into RNA, and remember the DNA is in the nucleus of eukaryotes and in the cytoplasm of prokaryotes. Regardless of where the DNA is located, all living things have DNA. That DNA has to be made into RNA, and we talked about there's messenger RNA, and there's transfer RNA, and there's ribosomal RNA. So we're talking about messenger RNA at this point, which is the copy of the DNA message. We talked about a little bit about like this. If you own the Mona Lisa, you wouldn't want to give it out to your friends. So you want to make a copy of it, you know, and you can hand them out the gift shop version. And that's what's going on here. You only have one copy of DNA. You don't want to send your DNA out in a really harsh environment, like a frat party. You want to send out a copy of that DNA, which is RNA. And then the RNA, in turn, goes out of the nucleus into the cytoplasm C. And we talked about the ribosomes, and you guys got tested on this. Remember, the ribosomes are the workers, and they convert the RNA into proteins, and that proteins are what makes us what we are, right? So flesh and blood. All right. So that's a review. That central dogma means DNA is converted to RNA, and RNA is converted into proteins. We talked about that before, so you already know that. You might not remember it, but you know it. Okay, so you know that inside your body, and we're talking about humans here. Thank you. We have 23 pairs of chromosomes, and you got 23 from your mom and 23 from your dad. And we talked about that the chromosomes, chromosome are made up of DNA, and those are wrapped around proteins that we call histones. So the proteins are called histones. They're kind of like the kite spools we talked about. The DNA gets wrapped around, and that's what allows us to package almost 8 feet of DNA inside the nucleus of a cell you can't even see with your naked eye. So we call the DNA and proteins chromatin, and long stretches of that are called chromosomes. So long stretches of DNA plus histones, the kite spools. So each gene is defined as a 4-letter sequence. Remember, DNA is, and we learned this in chapter 5, DNA is A's and G's and C's and T's And just like the letter of that English alphabet, the order of this matters. If I had the letters R-A-T, I can make rat or tar out of that, or art, and all those are different words. And then the RNA, again from chapter 5, is A's and G's and C's. And then in RNA, there's no T's, there's only U's. Again, this should be a review. And again, the order of these letters code for proteins, like hemoglobin, right, which carries oxygen in your blood from your red blood cells. So your whole genome we talked about is about around, scientists differ in this number, but somewhere between 20 and 35,000 genes make you what you are, right? And so when we're talking about this, you have a set of genes that you got from your father and a set of genes you got from your mother. And we talked a little bit about this in the last chapter. You know, you could get blue eyes. This is a terrible example because there's more than one gene for eye color, but I'm just going to use it because it's sort of mainstream. So let's say you got blue eyes from your mom, and you could get brown eyes from your dad. Right? So the gene is for eye color, and the variations are what we call alleles. So like, you know, so the gene would be, the gene itself would be the collection of the DNA that's get made into proteins that puts pigment into your eyeballs. And so the alleles could be blue, or they could be green, or they could be brown, and so on and so forth. So it's the variation of that. You know, we could say, okay, there's another gene, and that's for hair color, and the alleles for that would be blonde or red or brown or black, and so on. So that's what we mean by the word gene. It usually codes for something that you can see, like blonde hair. Okay, so this is not meant to be too busy, but it's just to show you that DNA, right? Here's a sequence of DNA. This is, I think I took this from alpha hemoglobin. Yeah, I did. So remember, the hemoglobin is 4 subunits, and if you forgot this, we talked about this in chapter 5, and there's 2 alphas and 2 betas, and together they make your hemoglobin, which carries oxygen to your cells through your blood. So this is the sequence, the DNA sequence for this subunit and this subunit, right? It's the same. So there's just 2 parts that you need to build this whole hemoglobin. Kind of think of it as a Lego set, right? So let's say you had 4 Lego pieces you needed to put together to make your Lego firehouse or whatever. So, you know, maybe these are blue Legos and these are red Legos. So they're the same, but you need them all together to make the structure. So here's the sequence. I underline this because all DNA, all genes start with this sequence, which is AUG in messenger RNA. And we'll learn about this. We're going to learn about this in chapter 17, when we talk about how genes are read and where they start and where they stop. But what I want you to see here is that there are 20 different amino acids, and there are 4 nucleotides, no matter if it's DNA or RNA. And that means that, like let's say, I don't want you to know these amino acids, but this is methionine, and this is valine, and this is leucine, and this is aspartic acid. And so they have just a 1-letter code. And so what I want you to see is that, let's say we assigned T to be M, and we assigned A to be V, and we assigned C to be L, and we assigned G to be S. Well, what can we assign to be P? There's no letters left. So we couldn't do that. So if in statistics, if you look at the possible variations here, we have 4 nucleotides. That means that there's 4 combinations of letters that we can have in one place. So one place is 4 combinations. And then we can do this to the power. So n, this is the number of places is n. So if we have 2 places, so we're talking about like T and A here, then we could do 4 to the second because it's 2 places, which would be 4 times 4, which is 16. So we would get 16 combinations out of that, but that's still not enough to get the 20 different amino acids. So we actually need 3 places, which is 4 to the third, and that is 4 times 4 times 4, which is 64. So now we have enough combinations to cover all the 20 different amino acids, which means that this has to be read in a 3-letter code like this AUG. And then the next one would be GUG and CUG and so on. And this is what we call the triplet code. So every one of these amino acids has a representative of 3 letters in either RNA or DNA. Any questions about that? Okay, so part of this chapter is to set you up for genetics. And so we're going to just define it here. Genetics is the study of heredity, which is things that you got from your parents, right? What's inherited from parent to offspring. But not only what's inherited, but the variation that is also part of that in a population, right? So if everyone had black hair, then that wouldn't be a very interesting genetic trait because there's no variation in a population, right? Even though you got black hair from your mom and black hair from your dad, if everyone had black hair and there's no variation, then the genetics wouldn't vary, and that would be really boring. So just to define this, heredity is the transmission of traits from one generation to the next. You probably already know what that means. And then variation is the differences that we see, right? Like brown hair and blonde hair and green eyes and blue eyes and so on and so forth. And this variation comes from variations in genes, right? So just to give you an example. So on a piece of DNA, I'm gonna make this up, but it doesn't really matter. So this is a chromosome. And we're gonna say on chromosome number 1, we have the gene for eye color. Now again, eye color is complex, so this is kind of made up, but it doesn't really matter. So let's say that this is also the number 1 chromosome. This you got from your mother. This you got from your father. Since I've already written down brown, we'll just say you get brown from mom. And let's say this one is blue and you get blue from dad, right? So if we looked at this gene, it would be made of A's and G's and C's and T's and so on. I'm just going to make it up. And so let's say that this gene is going to be turned into, so this is our gene, and this is going to be made into RNA. And I'm not going to go through the whole, A pairs with U and RNA. I guess I could. So in RNA, this would be U, and this G's pair with C's, and this would be G, and so on and so forth. And then that RNA will be read by the ribosomes, and that will be made into a protein. And that protein is going to fold into its shape, like we talked about in chapter 5, and that is going to reflect, like we talked about in chapter 10, that's going to reflect things that are in the brown wavelength. So whenever you look at someone's eye, it's brown because they're absorbing all the other colors except for brown, which they're transmitting to you, so that to you it appears brown, just like if you were wearing a red shirt, it would be absorbing everything but red and transmitting red. So what happens is that when it, like, let's say some radiation, could be uranium or something, could be a dentist office, it hits this piece of DNA and screws it up and turns it into a gene. All right. So when it does that, the protein doesn't fold in the right shape. Let's say it doesn't even fold, fold right at all. So let's say it's supposed to be like this, and it ends up looking like this. Well, this is not going to reflect light in the brown wavelength. And what it does is it's screwed up. So it's not going to reflect any light, and it's going to go to the default pathway. So think about it this way. If you move into a house or an apartment, what color are the walls by default?
They're white.
They're white, right? So if there's the same thing in nature, right? By default, your eye color is blue. So if you had no pigment at all, your eyes would be blue. In fact, if you have brown eyes and you want to make blue eyes, you could just go, they make a laser that will erase all the pigment out of your eyes, and guess what color your eyes are with no pigment?
Blue.
Blue. So you could make your brown eyes blue if you wanted to. And that really exists. And it's the same thing with blonde hair. So blonde by default is the lack of pigment, which is like a white wall. So if you have blonde hair, it's because you had, I mean, I would guess that most people started off with black hair. And it has to do with the protection for, black absorbs light. So it helps protect the DNA because light can damage DNA. So if you have blonde hair, it's probably because at some point in your past, your parents got a mutation in their brown hair gene that made it not fold right. And so by default, it doesn't make brown or black pigment, then your hair is blonde. So that's what we're talking about with variation. And so genes are generally broken in some way. So like if it's supposed to be, if you would normally make brown color, if it's broken, it goes to blue. If your hair is normally brown, if that gene's broken, it goes to blonde, and so on and so forth. All right, so one thing I want you to take from this is that this dirty word evolution, right? So evolution means, it simply means change in allele frequency. Remember, alleles. So let's say if we looked at all the population of people in Africa. So what color would you think that their eyes and hair are in Africa? The majority of people.
Brown eyes, brown hair.
Yeah. And then let's look at the population in Sweden. So what do you think their eye and hair color is?
Usually more blonde.
Blonde, right? And blue eyes. And this has to do with evolution, right? Because brown pigment absorbs light, right? Because it's dark. And so if you're in an area where you get a lot of light, you want the pigment to suck that light up so it protects your eyes. So it has to do with the amount of light that you're exposed to and how close you are to the equator. That has to do with how important it is that if you have brown skin versus light skin, or brown hair versus blonde hair. But evolution simply means that populations change over time. If you look at, there's a really cool movie on PBS. It's kind of hard to find because they used to give it out for free, but they got stingy. And it's called The Secret Life of Dogs. And so if you guys troll around on the internet, you might find a way to watch this. But it talks about how all dogs came from wolves, right? Every single dog, and it's not disputed at all, came from a wolf. And so what evolution says is that, well, do the population of dogs look the same? Like, does a Chihuahua look like a, I don't even know how to spell that. Does a Chihuahua look like a wolf?
No.
Does a Great Dane look like a wolf?
No.
Then they've evolved, right? Like, 200,000 years ago, most people had brown hair, and because most people lived in, around the equator, because everything else was covered in ice from the ice age. So the majority of the population had brown hair and brown eyes. And over time, that changed. So the blonde hair and blue eyed gene became more fixed in the population because they didn't live close to the equator, so they didn't need brown hair and brown eyes and dark skin. And that's what evolution means. Change in the frequency of the allele. Remember, alleles are like brown hair, blonde hair, and so on over time, which, and when we say time, we mean generations, right? So from one generation to another. And that's what evolution is. And evolution is simply a fact of life. I mean, I could simply say, do you look like your parents? Like 100%? If you don't, then you've evolved. All right. So we'll talk more about evolution when we get to the next chapter. But I want to kind of throw that out there. And we're going to talk a little bit now about a disease called hemophilia. So hemophilia is a disease it's a gene in the DNA, and it codes for a protein called factor. There's a few of them, but the main one is called factor VIII, right? And that's located on, remember we talked about sex chromosomes previously, and then we have an X and a Y if you're a male, and an X and an X, two X's if you're a female, right? So female, male. Anyway, this gene is located on the X chromosome. So this gene is on the X chromosome. It codes, it's DNA, so it's gonna make RNA, like we said in Dogma, and that's gonna make a protein. That protein is called factor VIII, and its job is to clot your blood. So if this gene is broken, what happens? Your blood doesn't clot. So if you had hemophilia, you had a defect in factor VIII, and you went outside and you were running around and you tripped and you fell and cut yourself, what would happen?
You would bleed to death?
You would bleed to death. That's exactly right. Not anymore, because we know that this is a protein. It's kind of like insulin. You can inject it into yourself. So if you have hemophilia, you could carry around like an EpiPen, and you could inject yourself with factor VIII if you got a cut, and then that would artificially clot your blood, because you're just putting the protein itself into your blood, like insulin. But back in the day, you'd bleed to death, right? And if you died, and then you wouldn't grow up to be, you wouldn't go through puberty, so you wouldn't be able to reproduce if you're dead. Makes sense? And so this gene would go down in the population, right? Because it's a bad gene. All right, so there's a term right here that I want you guys to know. It's called the locus, and it's actually short for location. So the location of the gene on the chromosome is called the locus. So the specific place where that's located. Does that make sense?
Yes.
Okay, cool. All right. So part of the extra credit for the next exam is for you guys to do a pedigree. So I go more into detail about pedigrees in the next chapter, but just so you know, so in a pedigree, this is the pedigree of the British royal family. So this is Queen Victoria, this is Prince Albert. This line here is a double line. That means that they're consanguineous. They're actually cousins or closer. So you can see that Albert's father, so squares are males and circles are females. And so Albert's father and Victoria's mother were brother and sister. And so they had children, and those children were cousins, and they interbred. This is pretty common back in the day because they wanted to keep all the land and the money in the family. Anyway, so they had a kid, and this is why inbreeding can be bad. So Victoria had a mutation. That's what this dot means. So this means that you're a carrier. And a carrier means on a genetic level. So we know Victoria is a female, so she would have 2 X chromosomes. Make sense? And Albert would be X and Y. This is a male. This is a female. And so on this chromosome, right, she has a copy of factor 8. And this is at a specific location, so we call it a locus. And so on one of these chromosomes, that is broken. So I'll just highlight it in yellow, as we'll say. That one is broken. And so, but it doesn't affect her because she's still making factor 8 from her other X chromosome that's perfectly fine. So this one is no factor 8. It's broken, but this one can make factor 8. So she gets half the amount of factor 8, but that's okay because it just makes your blood clot slower, but you're not going to bleed to death. That makes sense? And because she has one good copy and one bad copy, she's considered a carrier. It doesn't have to be something that's deadly. Like, let's say that the dominant trait is on the thing is brown eyes. And, you know, you let's say this is a number 1 chromosome. So you would have a brown and a blue. That would mean that you're a carrier for the recessive trait of blue eyes. And if you drew a pedigree and you were a female, you would make a circle and put a dot in it, and that could represent that you have 1 brown eye and 1 blue eye, right? You're a carrier. Now, if you had 2 blue eyes and you're a female, then you would shade it in. That means that you have the condition, the recessive condition. Here, where it's fully filled in, that means that they have the condition. This is a carrier, right? They have 1 copy of a bad gene and 1 good. This one means they have both bad copies. But what makes this unique is that in males only have 1 copy of factor 8. So if this one goes bad, right? If it's broken, Y can't save it. Which is what we call a sex-linked disease, because if you look, only males get this. You can see all the squares are males, remember? So all throughout here, it's all males that have this disease, this hemophilia disease. Females are usually just carriers because they have another copy, right? So even if they have the mutation, and then all these double lines here, these these are the inbreeding events. This is inbreeding. This is inbreeding. This is inbreeding. This is inbreeding, and so on. So this is why it can be bad to have inbred, you know, dogs or cats or whatever. And then people, it's bad to have inbred people, and that's why there are laws against, you know, marrying your first cousin or brother or sister or whatever to prevent this, because this can get expensive. Like medical care for the state can get really expensive for taking care of these recessive diseases that get fixed in the population. All right, so does everybody understand the pedigree? So these are the parents, right? They're breeding events or parent, you know, off mating events are horizontal lines, and then their kids branch off to the vertical lines, like here and here and so on. So they had a lot of kids. And the reason that these males get this is, we can kind of go into the genetics a little bit. And since this slide's up, I'm going to do this. So let's say Albert is perfectly normal, right? And he's going to, this is his DNA, right? This is Albert. And Victoria, we know she has 2 X's, and one is broken. So whenever she goes through meiosis, she's only going to package half of her DNA, right? Because your sperm, you want 23 chromosomes in sperm and 23 in an egg. So when they get together, they give you 46, which is the normal number of chromosomes. So that means you have to half your DNA. So Albert would package this, could package this as a sperm, or this is a sperm, right? Because he's a male, that's all he makes. And Victoria could package this as a egg, or this is an egg, and any sperm could fertilize any egg. So if this sperm fertilized this egg, then the result would be a person that got X from Albert that worked fine, and then got a broken X from Victoria. So this would be a carrier, and we would represent this person with a circle because it's female and a dot inside. Now let's say that, you know, this sperm fertilized this egg instead. So in that case, we would have an offspring. Let me erase this. In this case, we would have an offspring that had a good from Albert, good X, and a good X from Victoria. And so we would represent this by just a circle, no filled in or nothing. This is a perfectly normal female. And this is a carrier female. All right, so then I'm going to switch to blue. So let's say the Y sperm fertilized this egg. Well, what would we get out of that? We would get an X that was broken from Victoria and a Y that was fine. So this would be this person has no copies of factor VIII, and they're a male. So we would have to fill them in because they would have hemophilia, right? They'd have no way to clot their blood. So this is a male with hemophilia. And then we could say, okay, well, what if this sperm fertilized this egg? Well, then we would get a normal X, a normal X from Victoria and a normal Y from Albert. And so this would be a normal male. And we would just draw that as just a regular box. So this is a normal male, this is a normal female, This is a carrier female. This is a male with hemophilia. And we could get all those possibilities, and you can see from their kids, they got, this is a random occurrence. Any sperm can fertilize any egg. So they had normal males, they had carrier females, they had males that had hemophilia. And anytime you have this mating, you could have all those possible outcomes. Here, normal, carrier, or have the disease. And this is actually, you guys heard of Anastasia and the Bolshevik, the Russian Revolution?
Yeah.
So that actually, so one of these is Anastasia. The Russian Revolution was caused by, so back in the day they wanted male heirs, right? This is the offspring of Tsar Nicholas II, who was the ruler of Russia at the time, and he had 4 daughters, which were circles, and 1 son, which is here a square. And you can see his son Alexei was heir to the Russian throne, but there was a problem. What's the problem with Alexei?
He's hemophilia.
Yeah, he has hemophilia, and that means anytime he falls down and cuts himself, he could bleed to death. So there's this guy named Rasputin. I don't know if you heard of him or not, but he's kind of an evil guy. And what he did was he told the Tsar that he would make sure that his son survived to be the heir if he transferred all power to Rasputin. So ultimately, that it was trying to save this child, Alexei, that caused all the revolution. So in the end, if you want to boil it down, a genetic disease called hemophilia started the Russian Revolution and caused Anastasia and all these other people to be beheaded because they were trying to save this kid who had hemophilia so that he would be the heir to the Russian throne. So that's I find that kind of interesting, and I thought I would pass that on to you guys. All right. So hemophilia shows how that genes are passed on within families, right? And I kind of went over like how you can inherit this. It also shows that variation is important, right? If everybody, so let's just say everybody had hemophilia, right? The females to get hemophilia would have to have 2 broken X's. It's possible, right? And let's say the males had hemophilia, right? So if any of these, let's say if any of these sperm fertilized any of these eggs, every single person that came from that would have hemophilia. You guys see that? Because that's all there is. It's all broken genes. Here, here, here. So there's no possible way to get a good factor 8. So any offspring from this mating, they would all have hemophilia, no matter if they were a male or a female. So variation is important. Breeding within families reduces variation, and so that retains bad genes. It could also retain good genes, like let's say that you got flight. So if you bred back to your mom or your dad or whatever, then your family, like hemophilia, you could make sure that gene came out, even if it was a good, regardless if it's a good or a bad gene. And we know this with domestic animals, right? Like cats. So these inbreed cats, and if you're a breeder, a cat or a dog breeder, or even a horse breeder, you have to be real careful that you don't that you try to outbreed the bad traits and inbreed the good traits, right? Like if you're making a thoroughbred horse, it's super dangerous because even though you're going for good traits, like a really fast horse, these bad traits can follow along as well. So you get all of these really crazy diseases in these line of certain animals, and breeders try to avoid that by outbreeding their animals, or they should. All right, any questions about that? Okay, so just, we already covered this, but just to remind you, somatic cells are all cells in the body, and we talked about this in chapter 12. So mitosis, its job is to make somatic cells, right? A somatic cell is any cell other than a sex cell, which we, in humans, we're talking about sperm and eggs. All human cells contain 46 chromosomes, 23 pairs, right? 23 you got from your mom, 23 you got from your dad. And we covered this before. So this should say that chromosomes in somatic cells can just be distinguished by the size, the position of the centromere. Remember, centro is center and meros is part. So that's the center part, right? And remember, we have chromosomes. Let's say this is chromosome number 1. And you have 2 of those, one you got from your dad and one you got from your mom. And they'll have genes on there. And let's say that this is brown hair. And let's say this is blonde hair. So these, remember we talked about this, these are called homologous chromosomes because they're the same number chromosome. Like this is number 1 here. So you have one from mom and one from dad. And when we go through S phase, which we talked about in the last chapter, which is synthesis, you get a copy. So this DNA gets copied, right? And that copy is going to be held together by the center part, the centromere. And because these are identical copies of each other, they're called sister chromatids. So sister chromatids are different than homologous chromosomes. And you need to learn this because it's super critical when you're talking about genetics. There's a big difference. So sister chromatids are going to be identical. So that means that if this is brown, this would also be brown because it's a copy of it, right? The homologous chromosomes could be the same, but not all the way down the whole chromosome. You know, there's roughly 1,000 genes per chromosome, so chances are they're not going to be all identical. But you could have one like brown on one homologous chromosome and blonde on the other. So they're similar, but not the same. Does that make sense? All right. So make sure you know the difference between sister chromatids, they're exact copies, and homologous chromosomes, where they're similar, but they're like the same number of chromosomes. So these, one from mom, one from dad, those are homologous. You have a homologous number 2, 3, 4, 5, 6, and so on. And then these were supposedly numbered in order when they looked at them under a microscope, but in the end, I don't know if you can see this or not, but 21 is actually smaller than 22. So they screwed this up, but it's still stuck. So number 21 is actually the smallest chromosome. And then they got number 1 right. Number 1 is the biggest. So they're supposed to be numbered from biggest to smallest. So anyway, their size, right? Biggest to smallest, position of the centromere, where that center part is located. And then you can stain these things. They use a staining technique. It's called FISH, which stands for fluorescent in situ hybridization. You don't need to know that, but it causes it to have these bands, and you can see pink and blue and so on and so forth. So you can recognize the chromosomes from one another based on size, position of centromere, and banding pattern. This is what's important, and this is what I'll ask you on the test. Along with what's a somatic cell, what's a sex cell, how many chromosomes do we have? How many do we have from our mom, and how many do we have from our dad? What's a homologous chromosome? What's a sister chromatid? And that sort of stuff. I'll ask you that on the test. So make sure you know that. This is just what we just talked about, so we'll skip it. All right, so these are terms you're going to need to know. Gametes refer to sex cells, right? So gamete is a fancy name for, in humans, like a sperm or an egg. Diploid, ploidy is the number of sets of chromosomes we have. So we got 23 from mom and 23 from dad. So we have two sets, right? When we say a set, that's 23 is one set, and from mom, and 23 from dad is the other. So we would say that our chromosomes are 2n, because we have 2, and then the number is 1. This n number is 23, which is what n represents. So 2 times 23 is 46. So 2n, since n in humans is 23, 2n equals 46. All right. And that represents a term called diploid, right? So gametes are haploid. Now I know that this is a different word. We might say uniploid or whatever, but they just use haploid, and this is just how scientists talk, geneticists talk. So you're going to need to know that word. So haploid means 1n, right? And since n is 23, 1n equals 23, because 1 times 23 is 23. So our 1n is 23, and this is what we would have in a sperm or egg, right? Because we want the sperm to have 23 and the egg to have 23. So when they fuse together, they make a fertilized egg that has 46 total, which is the normal number. So, I might ask you, how many chromosomes in a human gamete? The answer should be 23, right? Because it's haploid. It's 1n. And I might say, how many chromosomes are in human somatic cells? And you would say 46, right? Well, is it haploid or diploid? Well, somatic cells are diploid because they have both the set that you got from your mom and the set you got from your dad. So they're 2n. Does that make sense?
Yeah.
Okay. So I know there's terms, foreign language, we talked about that, this part of this class. Fertilization, you probably know, that means when the sperm meets the egg, right? Sperm and egg fuse together. They form something called a zygote. So this word is important because we're going to talk about this in genetics. We use the word homozygous, and that means you have the same genetic makeup in the zygote, or heterozygous, which means you have a different genetic makeup in the zygote. So this word zygote is going to come back into play in genetics. So make sure you know zygote. It simply means a fertilized egg. A zygote is just a fertilized egg. So, and it's made from a sperm and an egg fusing together. So here's a sperm, here's an egg, they fertilize, they make a zygote. This is N, right? Sperm and eggs are 23, so they're haploid. This is 46, so it is diploid. And then this divides through mitosis into a full-grown human being. And then, if you're a male, then your testes make sperm, which are haploid. And if you're a female, then your ovaries make eggs, which are also haploid. And then those fuse, and then you have kids, and then they go through the same process, right? They go through mitosis, they develop, these sperm and eggs are made through meiosis, whereas the fertilized egg is made through, the human from the fertilized egg is made through mitosis. The sex cells are made through meiosis. So that's basically just the human life cycle, right? So you came from a fertilized egg from your parents, you grew up, depending on if you're male or female, you make sperm or eggs, and then you could have your own kids that would be made from a fertilized egg, and they would grow up, and then they would have kids from the sex cells that they made, and so on. So I don't really care if you know this. Some organisms have a different life cycle, but we're just going to focus on this one right now. So the life cycle is 2N to N to 2N, and so on and so forth, right? This is two different ends fused together, sperm and egg, to make another offspring. And remember the word zygote, right? Zygote is a fertilized egg. All right, so if you know mitosis, you kind of know meiosis. Meiosis is a little different because in mitosis, our goal is to be 2N, right? We want all the cells that come from the other cells to also have 46 chromosomes. In meiosis, we want it to be N, right? So we want it to have 23 chromosomes, because the sperm and the egg have to have half the number, because when they come together, they need to have the normal 46 number. So the other thing about mitosis is we want them to be genetically identical. Because if they're not, then that would cause mutations that could lead to cancer that we talked about. Here, we don't want them to be genetically identical, because we want to have variation, right? Like, think about it this way. If there wasn't variation between humans, that, like, say some humans are hairy and some humans aren't, and the next ice age, if we didn't have any hairy genes, everyone would be dead because they'd be frozen to death. You know, and of course we know how to, like, skin an animal and use that for clothes, but if we didn't, right, then, and we didn't have that variation, everyone would be affected by it. Same thing with HIV. So in HIV, some people have a mutation that doesn't allow the virus to get into their cells, and so they're immune to it, right? And that actually is left over from the bubonic plague, because the bubonic plague uses the same pathway as HIV to get into the cells. So the mutation that some people have, right, would protect them from HIV. Now, we're in the modern era where we know about viruses and stuff, but think about what would happen if HIV came out in the Middle Ages and nobody had the variation to make them immune to it. In the end, the human population would be completely dead. So we need variation, right? It helps us survive as a species. And that's the goal. In meiosis, we want them to not be genetically identical so we can get variation. Here, we want them to be genetically identical. These are only sex cells. These are somatic cells. All right, so this is what's going on. Remember, we talked about the cell cycle, and we have S and G1 and G2 and M. So we're talking about M phase. This is interphase, right? Remember, interphase is everything besides M phase. So in interphase, let's say this is dad's. We're just going to call this a #1 chromosome. This is dad's #1. This is mom's #1, right? And it's going to go through S phase, and it's going to make a copy, right? And it's going to get held together by the centromere. And so now mom's chromosome got copied. I guess I should make that blue. That's not even blue. Mom's chromosome got copied. Dad's chromosome got copied. Dang it. And that's what it's showing here. Right, so if there was blue eyes on this one, both of these would have blue eyes. If there's brown eyes on this one, both of these would have brown eyes. And these are called sister chromatids because they're genetically identical. They're copies from S phase. Make sense? All right, so here's the difference. Remember in meiosis, and you guys did, or mitosis, when you guys did this. So let's say that this is number 1 from mom. Oh, that's dad. This is number 1 from mom. Mom. Let me erase this real quick. What are you doing? Oh. Whatever. Go away. All right. So in mitosis, these sister chromatids lined up on the metaphase plate, and they got divided right down the middle. That way, you were sure that you'd get number 1 from mom in one cell, right? Number 1 from, I'm sorry, this is dad. Number 1 from dad, and number 1 from mom. We go to this side, so these right here. And then these other ones, number 1 from dad and number 1 from mom, would be in a different cell. So that we would have genetically identical ones. But here, it doesn't line up like this. They line up as homologous chromosomes. So instead of being on a line, they line up like this together. And we call that a tetrad because there's 4, right? These are sister chromatids. They're together, they're homologous chromosomes. This, because there's only 2, this would be called a dyad. So they line up as dyads in mitosis and tetrads in meiosis. And these divide down the middle. But here, I want you to notice this is different, right? All of the chromosome material here is from dad. And all of the chromosome material here is from mom. That's different than this, right? Because it's divided right down the middle. And so this is both mom and dad. Mom and dad. Mom and dad. That's different here. And then it goes through a second round, which is exactly identical to mitosis, where it divides down the middle. And then we get sex cells with half the DNA, right? And you can see that they're different from one another. Does that make sense? Maybe, maybe not. All right. So I'm gonna show you a video, and then we'll look at it again. And it's gonna look at the difference between meiosis and mitosis.
Your car collecting dust? Share your car on a bill and turn that dust into cash. We make your car sparkling clean and sanitize it before and after every trip. Get more out of your car and more money for the things you want. Avail. Share your car and earn today. In biology, there are often vocabulary terms that sound pretty similar. Chromosome, chromatid, chromatin, transcription, translation, mitosis, meiosis. You probably have encountered this. When I was first learning about mitosis,
Can you guys see this?
I learned them both separately first.
Yeah.
There.
Think about them, what was different, why did they both matter? I would try to compare the stages by flipping through images. You know what would have really helped me? A side-by-side comparison. And that's what this video is. We assume you already have a background of mitosis and meiosis. If not, take a look at our videos on them. But this video is a side-by-side comparison, presented in a split screen. Mitosis on the left, meiosis on the right. Both of these processes, along with the cytokinesis that follows them to split the cytoplasm, are involved in making new cells. Mitosis results in body cells. Meiosis results in sperm and egg cells, otherwise known as the fancy term. Before we start mitosis and meiosis, let's look at what you start with. Your starting cell in both mitosis and meiosis is diploid. Written here is 2N. That means it has 2 sets of chromosomes. In humans, that's including 1 set of 23 chromosomes from mom and 1 set of 23 chromosomes from dad. 46 chromosomes total. During interphase, the cell duplicates the chromosomes. When you duplicate 46 chromosomes, you still say there are 46 chromosomes, as the newly duplicated portion is still attached at the centromere region. But there are actually 92 chromatids. Interphase isn't part of mitosis or meiosis, but it's a really important phase because it duplicates chromosomes before we get started. Just to point out, it's really hard to draw 46 chromosomes. We're going to use 6 chromosomes in our diagrams when we illustrate what's happening, because it's much easier to draw and visualize. Oh, and just a fun fact, some insects have 6 chromosomes, like mosquitoes. Unfortunately, I'm not a fan of mosquitoes, but mosquitoes do both mitosis and meiosis. When learning the stages, we get the acronym PMAT, which is helpful for understanding the stages. Both mitosis and meiosis go through these stages, but meiosis goes through them twice. And
Okay, so I just want to point this out that your book is a little different here. So your book has a pro-metaphase in mitosis, which is an extra step that it doesn't talk about in meiosis. But other than that, what they're saying is the same. So just keep in mind that there's an extra stage that you guys learned between prophase and metaphase and mitosis.
therefore has a number next to each PMAT stage. We're going to show some basic events for each PMAT stage, but please know that there is way more detail to explore than what we can include in this quick video. Prophase in mitosis. Remember that pro can mean before, and this stage comes before the others. The chromosomes are visible. We say they're condensing, which means they are thickening. Prophase 1 in meiosis. Happening here too, but the chromosomes are actually going to match up with their homologous pairs. The word homologous means that the chromosomes are approximately the same size and they contain the same types of genes in the same locations. With each pair, one came from mom and one came from dad. In this formation, chromosomes can transfer their genetic information and exchange it between each other. It's called crossing over. It can make for what we call recombinant chromosomes. Metaphase in mitosis. The nuclear envelope which had surrounded the nucleus was already disassembled before metaphase even started. For metaphase, I like to remember the M for middle, because in this stage, the chromosomes line up in the middle of the cell in a single file line. Metaphase 1 in meiosis. The chromosomes are in the middle as well, but they're still going to be in pairs in the middle of the cell, so it's not a single file line. Anaphase in mitosis. I like to think of the A for away. The chromatids are pulled away by the work of the spindles. They are moving to opposite sides of the cell. Anaphase 1 in meiosis. Same thing, but in this case, it's the chromosomes, not chromatids, being pulled away to opposite sides of the cell. Telophase in mitosis and telophase 1 in meiosis. The chromosomes are actually the complete opposite ends, and new nuclei are forming on each side to make 2 new cells. And they are starting to surround the chromosomes on both sides, as this will eventually form 2 cells. Cytokinesis follows to split the cytoplasm to complete the actual dividing of the cell. So at the end of mitosis and cytokinesis, we end with 2 identical diploid cells. In humans, they would both have 46 chromosomes. This is great for organism growth. Growing requires making more cells after all, or replacing damaged cells. Onto meiosis 2. Prophase 2. Chromosomes are condensing in both cells. It's not going to be as eventful as it was in prophase 1 because they're not going to have homologous pairs and crossing over. Metaphase 2. M for middle, but this time the chromosomes are in a single file line, similar to how metaphase looked in mitosis. Anaphase 2. Think A for away. This time, though, it's actually the chromatids that are getting pulled away. Telophase 2. Chromosomes are at the complete opposite ends, and new nuclei are forming on each side to make these new cells. Cytokinesis will follow meiosis 2 to completely split the cytoplasm. We are now finished with meiosis, and we end with 4 non-identical cells. Gametes. Males make sperm cells in meiosis, and females make egg cells in meiosis. These gametes are haploid, meaning they have half the number of chromosomes as the original starting cell. In the case of humans, the resulting cells would each have 23 chromosomes. By the way, when a sperm and egg cell combine, it results into a diploid cell, a fertilized egg, otherwise known as a zygote, which will then start a series of divisions using mitosis to give rise to a brand new organism. Well, that's it for the Meiosis sisters. And remind you to stay curious.
Okay. Any questions about that? So I wanted to kind of show you that because it's sort of animated and you can kind of see. But remember, in mitosis, the goal is to get genetically identical cells, and there's only one cell division. In meiosis, there has to be two cell divisions. So the first one we would get 46, and then the second one we would get half that, which would make the sperm and the egg with 23. So I'm gonna go jump back over to the lecture, the PowerPoint. All right, so we're gonna go through each of these steps that you just saw in the video, but this is for meiosis, not mitosis. So it's a little different, but the same sort of things happen. So in interphase, remember that's everything but that's so here's our G1, S, G2, M, and interphase is everything but M phase. So that is interphase. And in interphase, the DNA is replicated, right? So we still have S phase. We have 46 chromosomes here because it comes from a normal cell. Meiosis is made by cells that were derived from the joining of a sperm and an egg. Like, so they all have all the cells in your body have 46 chromosomes to start with. S phase, we're going to double that, so we get 92. And then it's going to go through meiosis 1, and then the end of meiosis 1, we're going to half this, so we'll end up with 2 cells with 46, and that's still too many, right? So we're going to go through another process called meiosis 2, which will half these, and we'll end up with 4 cells with 23 in each. And this could be sperm if you're a male, or it could be eggs if you're a female. So in interphase, it precedes meiosis, just like we showed. The chromosomes are replicated in S phase from 46 to 92. When they get copied, they're joined together as sister chromatids, remember, at the centromere. And then the centrioles, when you have, so you have the 2 centrioles that we talked about, right? They make the microtubules and they attach. When the cell divides, it only ends up with 1 centriole. And so in order to make those 2, so it can, pull the, like I told you, can you pull the pen cap apart with 1 hand? Most people are like, no, I need 2. So this has to get duplicated and then it moves over so that it can make a new one so that these cells can also divide. So the centriole replicates to form this spindle, and so it's just talking about this making a copy of itself, and they're going to start migrating. Notice the nucleus is still intact. In meiosis, we only have prophase, metaphase, anaphase, and telophase. So there's no pro-metaphase. Now, some interesting things they talked about in the video. We know the chromosomes condense. They have to because they would get all tangled up like headphones in a gym bag, and they'd get ripped apart, and that would be bad because they could break in the middle of genes that are really important, like for glycolysis, and then you would die. So you don't want them to get tangled up, so they condense. Remember, that just means that they're wrapping tighter around those histones. There's something called synapsis. So in the human language and in genetics too, there's lots of names for things that society thinks is really important. So you can think of like, how many different words can you think of for money? Money, bones, dice, dough, dinero, and so on and so forth. So this is a really important thing called synapses. And what it is remember they line up as tetrads, right? So we have sister chromatids. Let's call this the number 1 chromosome. And so mom, this is dad. He's got a copy of his chromosomes and mom has of hers. Now, remember in mitosis, we have the centromere to hold it together. And we want to, and the purpose of that is so that they all move together. So that when they line up, we know they're lined up as a pair. It's kind of like the buddy system with swimming. Here we have 2 different ones, but we want them to move together so that they line up together, so that we get number 1 lined up and number 2 lined up on the metaphase plate. Remember, it's going to be shorter, and number 3 lined up, and that's going to be shorter, and so on. So we want them all lined up in the middle, right? But they wouldn't line up like this unless we had a way to connect them. And so the chromosomes actually cross one another, and in doing so, they've part of dad's chromosome ends up on mom, and part of mom's chromosome ends up on dad's. And you can see that here. So we call that synapsis. It's also known as crossing over, right? So there's another term for it, or recombination or homologous recombination. So for this class, you need to learn, because this is how they talk, you're going to learn that this exchange of genetic information to hold these tetrads together as 4s is called synapsis, crossing over, or recombination. So make sure you know all 3 of those. And that's where genetic material is exchanged as well. So not only does it hold all these tetrads together, but because we get some of mom's chromosome on dad and some of dad's chromosome on mom, I'm just going to draw this as blue so you can see. So some of this chromosome here is going to end up on this one, and some of this chromosome here will end up on this one. And so that increases variation, which is our goal, right? In meiosis, we want to make it as much variation as we can and half the amount of DNA. And so this is prophase 1. The nucleus comes apart. We have this crossing over event that it only this only happens in meiosis, not mitosis, and we form tetrads. So nucleus breaks down, centrosomes move apart. It's the same as mitosis. And then I'll talk about metaphase, and then I'll end it here. So in metaphase, remember, there's a bunch of pushing and pulling going on to line these things up on the metaphase plate that we talked about in mitosis. But here they line up as tetrads, where in mitosis they lined up as sister chromatids. So we'll pick this up on Tuesday next week. I don't have office hours tomorrow. I have a department meeting. So if you guys have any questions or whatever, you can hit me up today or Monday during my office hours. And then I'll pick up the lecture, and we'll probably finish up this lecture on Tuesday. All right, so any questions?
Just to recap this chapter 13, this part of the exam, right? The chapter 3.
Yes, so on exam 3, which is coming up, you're going to have chapter 10 12, and 13.
And that closes on 28th, right?
I'll have to check the exact dates, but yeah, I'll end up having to move that. I'll move that to open on the 28th and close on the 3rd.
Okay, just to plan. So
Yeah.
Synapsis, kind of like synchronized swimming. That's what I'm thinking of.
Yeah, kind of. I mean, you want to keep everything together, because if these floated around, you wouldn't know what chromosomes went where. So it's kind of like having all your kids hold hands, so you know that they stay together as a group.
Like tubing, that would be a good example, right? Like if you tied tubing down the river.
Yeah, sure. Yeah, because then, like, if you're not together, people could float away. You don't know where they're going to end up. So it's the same thing here. You want to make sure that all of these chromosomes that are the same or similar all stick together. That way you can make sure that they're equally divided into the new cell. And that's really the purpose of it. That's the purpose of a centromere, and that's the purpose of this crossing over, is to keep everything together, like holding hands or holding tubes. Any other questions?
Great, thank you.
All right, guys, have a good weekend. I'll see you next week.
Thank you.
All right, bye.